So in the last class we have studied what that how this glucose is converted to the pyruvic acid. Yes or no? This one. No? So can you tell me how many ATP is being released in what is the net gain of ATP over here?
So two.
Uh, so 2 ATP. How? Listen, see here. There is a 2 ATP is released. Another here is again 2 ATP. Okay, total is 4 ATP actually released, but there is a use of 2 ATP over here. Okay, 1 ATP here and 1 ATP here. So if you will net gain, you will be get to 2 ATP. Okay, and how many NADH? We'll go through here. See this one. See how many NADH over here? Here is how many NADH here? Two, na? Yes or no?
Again.
Yeah. Dita NADH ଆଉ କେତେ ATP ଦିଟା ATP ଓକେ ତ ଦିଟା ATP ଆଉ ଏ ଯଦି ଦିଟା NADH କୁ ଆମେ ଭାଙ୍ଗିବା ATP ରେ ତାହେଲେ 2 3 ଗୁଣନ 3 6 6 ଇଏ ପ୍ଲସ 2 କେତେ ହେଲା ଟୋଟାଲ ହେଲା 8ଟା ହେଲା no so if you will say anyone asks that the total number of ATP means substrate level there is 2 ATP and NADH data if you have to convert it into ATP then it will be 6 ATP okay fine. Now we'll study about what is the means how this glucose pyruvate will fate of pyruvate. What will happen to the pyruvate on the presence of oxygen and the absence of oxygen? Okay. So in the fermentation, say by yeast, is the incomplete oxidation of glucose is achieved under the uh, anaerobic condition by the sets of reaction where a pyruvic acid is converted to the carbon dioxide and ethanol. Okay. So what is saying that in fermentation said by yeast is the incomplete oxidation of glucose. Okay. What happens over here is there is a incomplete oxidation of a glucose is going on. Okay. Give me a moment, please. So the enzyme pyruvic acid decarboxylase and the alcohol dehydrogenase catalyze this reaction. So as it is a incomplete oxidation is happening. So here the catalyst, those who are catalyst are releasing the either the lactic acid or the ethanol. Okay, so one is pyruvic acid decarboxylase or alcohol decarboxylase or lactic acid dehydrogenase. Okay. So other organisms like some bacteria produce lactic acid from the pyruvic acid. Okay. The steps involved are the shown in the figure 12.2. This is the actual figure what is given over here. So in animal cells also like muscle during exercises, when oxygen is inadequate for the cellular respiration, pyruvic acid is reduced to lactic acid by the lactate dehydrogenase. This reducing agent is here NADH H plus, which reoxidates to NAD plus. So what happens over here? Sometimes what happens if you see the sprinter or those players who are continuously running and due to the shortage of oxygen this pyruvic acid cannot convert completely, it is not oxidized, so that it is due to incomplete oxidation what happen there will be lactic acid will be formed and lactic acid generally causes the muscle cramp in the athletes. Okay, so by how it happening it is by the enzyme followed by the enzyme lactic acid dehydrogenase. Or if in the case of yeast and if the oxygen is absent, then it either will be ethanol, okay, ethanol by the pyruvic acid dehydrogenase or alcohol dehydrogenase. Okay, in both lactic acid and alcohol formation, not much energy is released. Okay, so here that much energy will not be released. Okay, less than the 7% of energy in glucose is released, and all the trapped as high energy bonds of ATP. Also, the process are hazardous. Either acid or alcohol is produced. What is the net ATP that is synthesized? What is the net ATP we are synthesized over here? Now here, generally in the if you will ask that in this case in the if ethanol is releasing, then what is the net ATP we are getting? Now again, see that in the glycolysis 2 ATP, here there is no ATP synthesis is happening, same 2 ATP. Okay. What is saying that? What is the net ATP synthesized? To calculate how many ATP are synthesized, deduct the number of ATP utilized during the glycolysis. When one molecule of glucose fermented to the alcohol or lactic acid. Yeast poison themselves to death when the concentration of alcohol reaches about 13%. Now, when the alcohol is releasing more than 13%, then now the yeast having getting the death. Okay. So, what would be the maximum concentration of alcohol beverages that are naturally fermented? That you have to tell. Okay. So naturally fermented. So how do you think alcoholic beverages of alcohol content greater than the concentration obtained? How generally now in the that liquor whatever you're getting, sometimes you are getting 13%, sometimes 8%, sometimes 42%. Okay. So how it is being calculated, or how it is being beverages alcoholic content greater than this concentration are obtained. So how these are obtained due to the process of fermentation, how it is happening. So that he is asking the question. Okay. So when there is a process by which organism can carry out a complete oxidation of glucose to extract the energy stored to synthesize a larger number of ATP molecule needed for the cellular metabolism. In eukaryotes, these steps takes places within the mitochondria and required oxygen. So what happening is here that when the generally the when the large amount of energy to be released, here there is a presence of oxygen is required, and that the eukaryote cells will be involved. That aerobic respiration is a process that leads to a complete oxidation of organic substances in the presence of oxygen and release carbon dioxide, water, and large amount of energy in the presence of a substrate. Okay, so this type of respiration is most common in the higher organisms. We will look at this process in the next section. So now please go through over here what is he saying about the fate of this. Okay. Now here, see lactic acid aerobic respiration in human muscles during exercise when oxygen is inadequate. Alcoholic fermentation in yeast. So what is saying that alcoholic fermentation used in the formation of beverages, alcoholic drinks, bread. Okay. So glucose, glyceraldehyde, or 3-phosphate lactic acid. Okay, you just this is not correct actually. Something is copied. Okay. So the bread become puffed or spongy due to the release of carbon dioxide during the process. Whatever that you are saying that bread getting softness, so it is due to the release of carbon dioxide. In both lactic acid and alcoholic fermentation, not much energy is released. Less than 7% of the energy in the glucose is released, and not all of it is trapped by the high energy bonds of ATP. Also, the process are hazardous. Either the acid or alcohol is produced, yeast poison themselves to death when the concentration of alcohol reaches about 13%. When there is a release of alcohol and it is more than 13%, now yeast poison themselves to get death. Okay. So in anaerobic respiration or fermentation, the net gain of ATP is how much? 2 ATP. Sorry. Because during this process in glycolysis, 4 ATP are synthesized by the substrate level phosphorylation and 2 ATP are consumed. So net gain equal to how much? 2 ATP. Then the 2 NADH produced during the glycolysis do not enter into ETS. Instead, they are utilized from the alcohol to lactic acid. Now see over here. This is a diagram of that same diagram, a bit more 3D labeling is given. So what happened here, that there is a NADH is formed over here. See, there is 2 NADH is formed. Now these 2 NADH, either it will be used in the from the pyruvic acid to ethanol or the pyruvic acid to lactic acid. There will be use of NADH. So this NADH is already being used in the synthesis of lactic acid and ethanol. So how then what is the gain of what is how much number of ATP will be there? So it will be 2 number of ATP. Okay. So that's what he's saying, that in glycolysis 4 ATP are synthesized by the substrate level phosphorylation and 2 ATP are consumed in the first two steps. Okay, fine. Now see over here. Here, D type, 2 ATP. 1 ATP is here used, 1 ATP is here used. So total number of 2 ATP, 2 to 4 ATP. So substrate level this net again net if you count the net, then it will be 1 is over here is used, 1 is here is. Glucose to glucose 6 phosphate and fructose 6 phosphate to fructose 6 1 6 bis phosphate. Okay. So net gain is here how much? Not 2. And these two NADH is used in the where if it is the anaerobic respiration over here, if it is anaerobic respiration and release the lactic acid and the lactic acid or ethanol, then these NADH will be used over here. So there are total gain of a 2 ATP over here in the anaerobic respiration. Okay. Instead, they are utilized to form alcohol or lactic acid. Lactic acid fermentation is also performed by bacteria, lactobacillus, that is generally whatever you are using that milk to curd and these things. It is used in the curd, other dairy product formation. Curd become our sour due to the excess lactic acid formation. Okay. In anaerobic respiration, there is external final electron acceptor, that is oxygen, while in the fermentation, there is no external electron acceptor. The final electron acceptor is the organic intermediate of the process. Right? I think you understood this process. The fate of pyruvate means if the oxygen are not available. Okay, fine. We'll do the further now. Come to the aerobic respiration, or any doubts over here? Hello. Any doubts?
No, sir.
Okay, fine. Sorry. Now comes to the aerobic respiration. Okay. Now for aerobic respiration to take place within the mitochondria, the final product of a glycolysis. Okay, pyruvate is transported to the cytoplasm into the mitochondria. The crucial event in aerobic respiration, sir, there are generally two steps. Okay, one is what? There is a link reaction is followed, that is pyruvic acid to acetyl coenzyme. Okay, and that acetyl coenzyme will be it will get to the carbon dioxide, means completely oxidized in the that is called as the Krebs cycle. Okay, so it is saying that a complete how this crucial events of aerobic respirations are happening, and the complete oxidation of a pyruvate by the stepwise removal of all hydrogen atoms, leaving 3 molecules of carbon dioxide. Now, what is happening over here? The complete oxidation of pyruvate. Now, this pyruvate having the 3 carbon compound. Yes or no? First, the glucose is 6 carbon compound. If you have gone through, then this 6 carbon compound, in case of glycolysis, I'm saying the 6 carbon compound glucose is being converted into the 3 carbon compound pyruvate. Now, this 3 carbon pyruvate will be oxidized in the whole process of aerobic respiration in the mitochondria, and it will release 3 molecules of carbon dioxide. Means how many carbons is there? It will be completely oxidized during the nature. So now the passing on the electrons removed as a part of hydrogen atom to the molecular oxygen with the simultaneous synthesis of it. So what is interesting to note that the first process takes place in the matrix of mitochondria, while the second process is located in the inner membrane of mitochondria. So generally there are total number of a Krebs cycle if we will go through, there are total 8 number of reaction. Okay. So 7 number of reaction happens in the mitochondria matrix, and only one last one, not last one, it is from the I think succinyl coenzyme to fumarate. It's like that. So there is a succinase dehydrogenase enzyme is there, which is generally present in the internal membrane of the mitochondria. Now see over here. This is the structure of a mitochondria. Okay. Now this is the matrix. This space in between the membrane. Okay. This is matrix. Okay, mitochondrial matrix, and this lining is called as the inner membrane. Okay. This inner membrane, this succinate dehydrogenase is present. I will go through that. Okay. In the next slides, we'll go. Okay. Now see over here. Pyruvate, which is formed by the glycolytic catabolism of a carbohydrate in the cytosol. Now this line is how it is how it is constructed. See, pyruvate, which is formed by the glycolytic catabolism. Glycolytic catabolism means in the glycolytic pathway how it is being converted to the pyruvate. Okay. About how what the carbohydrates means of glucose in the cytosol. In the cytosol means in cytoplasm. So pyruvate, which is formed by the glycolytic catabolism of a carbohydrate in the cytosol, after it enters to the mitochondrial matrix, undergoes a oxidative decarboxylation by a complex set of reaction catalyzed by the pyruvic dehydrogenase. Now, in the first step, what is happening? This pyruvic acid is going to the oxidative decarboxylation. What is oxidative decarboxylation? Now, oxidative decarboxylation means that the addition of oxygen and removal of carbon dioxide in a same step is called as the oxidative decarboxylation. Understood? Because we will study that there are another two numbers of oxidative decarboxylation is there. So remember that oxidative carboxylation is what? The addition of oxygen and removal of a what? Carbon dioxide. Okay. Now what happened? This pyruvic acid, this is a 3-carbon compound. Okay. Why this coenzyme is given? Because the coenzyme is given for the to stabilize the energy release in the process. Okay, so pyruvic acid coenzyme है, okay, oxidized, okay, fine, oxidized in the presence of a pyruvate dehydrogenase. There is acetyl coenzyme A is formed and the carbon dioxide is released. Now see what happens. This 3-carbon compound is converted to 2-carbon compound and there is a carbon dioxide release over here. Now there is 1 carbon dioxide is released. Now how many carbon dioxide is released in the further steps? I'm asking you people. Then how many carbon dioxide will release in the next further step? 1 carbon dioxide is here released in the link reaction. This one is a link reaction. What you are seeing? This one is a link reaction. 1 carbon dioxide is released. We are saying that a complete oxidation of a what? Complete oxidation of a pyruvic acid or pyruvate will be there in the whole process in the aerobic respiration. 1 carbon dioxide will be released over here. And now how many carbon dioxide will release in the Krebs cycle? In 1 carbon dioxide release in the link reaction, how many carbon dioxide will be there to release in the Krebs cycle? That what I'm asking.
3.
ତିନିଟାରୁ ବା ଗୋଟେ ଗଲା ହାରି ଏଠି କ୍ରେବ୍ ସାଇକେଲ ଆଉ କେତେଟା ଦିଟା ଟୋଟାଲ ହେଲା so here this NAD is reduced and the process is oxidized. Okay, fine. Now this acetyl coenzyme A now will enter to the Krebs cycle. Okay, why this? Okay, why this is Krebs cycle? Because the first acceptor, okay, is a say the first product is a citric acid, and citric acid, if we will go through the structure of citric acid, there will be a three number of COH group is attached in that. So that's why you can say that tricarboxylic acid. Okay, so if we will go to the citric acid, okay, structure of citric acid. Let's see. Now see, there is 3 number of a COH group is added, carboxylic group is added. That's why we are saying that this is a tricarboxylic. Okay, understood? Now but this TCA cycle starts with the condensation of acetyl group that I will tell you. Just give me a moment. So what happened over here? This 3-carbon why it is tricarboxylic? I said about that there are 3 number of carboxylic group is there. Okay. One more thing is there. As it is given by the Krebs, so it is also known as Krebs cycle. Okay. Now this acetyl coenzyme A will go through the process and the first product is called as a citric acid. This is a 6-carbon compound citric acid. Okay, that we will see in the this slides PowerPoints. Okay, now over here come. Now, for aerobic respiration to take place within the mitochondria, the final product of glycolysis is pyruvate is transported from the cytoplasm into the mitochondria. Okay. Now, the crucial events in the aerobic respirations are now there is a complete oxidation of a pyruvate by the stepwise removal of the hydrogen atoms, leaving three molecules of carbon dioxide. Link reaction and the Krebs cycle. In the link reaction, one carbon dioxide is released, and the Krebs cycle, two carbon dioxide will be released. Now, passing on, the electrons removed from the part of hydrogen atom to molecular O2 with simultaneous synthesis of the ATP. Now, ATP will be released simultaneously. Now, this is the that you have studied the Krebs cycle where is happening. There will be in the matrix. Link reaction is also happening over here, the matrix. Now, come to here. So, link reaction is what? This process connect glycolysis and Krebs cycle. So, it is link reaction or gateway reaction, we are also saying. Okay. Now, during this process, first time carbon dioxide is evolved during the respiration. Okay. Pyruvic acid, 6 carbon compound glucose is converted to pyruvic acid. Now, this pyruvic acid, when it is going through the link reaction, first time CO2 is evolved during the respiration. Which respiration? Aerobic respiration. Okay. Now, if some question is asked that in what process or in what process first time the carbon dioxide is evolved during the aerobic respiration, that is link reaction. Okay. Now, acetyl coenzyme A is a connecting link between the glycolysis and Krebs cycle. Okay. So decarboxylation and dehydrogenation, or you can say that oxidative decarboxylation takes place during the formation of a acetyl coenzyme. Now, what I have said, pyruvic acid going under that I have said you before in the NCRT when I am discussing the NCRT that pyruvic acid is going through the oxidation on the presence of a what? See over here. On the presence of water. Okay. Oh no, sorry, this one. And then coenzyme, sorry, on the presence of this magnesium ion and this coenzyme A will be added so that it is going through the oxidative decarboxylation, and here the carbon dioxide will be released, and this NAD is reduced to NADH, and the whole the pyruvic acid is oxidized to acetyl coenzyme A and carbon dioxide. That's what he is saying in the this slide. Okay. This pyruvic acid, coenzyme A, NAD plus in the these are the cofactors. Okay, Mg plus 2. So pyruvate dehydrogenase, so acetyl coenzyme A, carbon dioxide, NADH plus. This is the link reaction. Okay. If you have understood the link reaction, we'll go to the next step. Okay. Here you have seen that there is a one reduction of NADH. Okay, in the link reaction. This you count on because okay. Otherwise you will forget. Now comes to the Krebs cycle. Now this cycle is discovered by the Krebs. Okay, it's a Krebs. So this tricarboxylic acid cycle occurs in the mitochondrial matrix. Where is the location? Remember mitochondrial matrix. All the enzymes of TCA cycle except oxidate dehydrogenase that I have already said before in the inner mitochondrial membrane. So here this oxidate dehydrogenase is present in the inner membrane. This is the membrane bound enzyme. Okay, this one. Okay, and rest are all are in the matrix. Okay. So Krebs cycle is also called as the citric acid cycle because is the first compound is a 6 carbon compound citric acid. In this acid, there is 3 carboxylic group are formed. So process is called as the TCA, tricarboxylic acid cycle. Okay. In Krebs cycle, oxaloacetic acid is the first member and is also the first acceptor of acetyl coenzyme. So who is accepting the acetyl coenzyme? Now oxaloacetic acid. This oxaloacetic acid is a 4 carbon compound. You know, in the time of C4, I think C3, yeah, C3 or C4, C4 plants. Okay. So in that, you remember how many carbon compound? Okay, oxaloacetic acid and malic acid, citric acid we have studied in that time. Okay. This oxaloacetic acid receives the so that what acetyl coenzyme to form the citric acid. Okay, the first product. So 4 carbon. This is 2 carbon acetyl coenzyme. So there will be 6 carbon will be formed in the citric acid. The PCA cycle starts with the condensation of acetyl. Condensation means water is added. Okay. Group with the oxaloacetic acid to yield citric acid. The reaction is catalyzed by the enzyme citrate synthesis. Generally the first enzyme is generally acts. This is the citrate synthesis. Now over here, see here. Now this is the oxaloacetic acid, 4 carbon compound. There is a acetyl-coenzyme 2 carbon compound will be added over here. Here the water is added, so that is done by the generally the process is called as the condensation. And this coenzyme A, this is acetyl coenzyme which is added over here. This one here is released. Okay. Now this product is first product. This is the first reaction. The first is citric acid. Okay. Now this citric acid forms a isomerase. Okay, isomer. Okay, that is called as a isocytric. Okay, this is okay. Then you just go. Don't. This generally water is whatever water is given here, water is removed. Then again it is okay. So please go to this citric acid. It is isomerase. Okay, this is isomerase activity is happening over here. Now this is isocytric acid. Now this isocytric acid again will go through the isocytrate is oxidized and the NAD is reduced over here. What happening here? Now oxalosuccinic acid. Now this is the first reaction. Remember oxaloacetic acid we added with the acetyl coenzyme A. Citric acid. This is the first. Citric acid isomerases into isocytric acid. Second reaction. Then this isocytric acid oxidized to form oxalosuccinic acid. Okay. Here one NAD is reduced to NADH. Now this oxalosuccinate will over here there will be again what oxidative decarboxylation. Okay. This oxalosuccinyl acid. Okay. There will be a what you can say that carbon dioxide will be again involved and that oxidation is happening. Okay. This is what alpha ketoglutarate acid. Now this again alpha ketoglutarate acid again going under the oxidative decarboxylation. Okay. To form succinyl coenzyme A. Okay. And again this coenzyme A is added. So these are the things. So from here oxalosuccinic acid to the succinyl A, okay, fast carbon dioxide is released in the link reaction. Okay, understood. In the then second carbon dioxide is released when this oxalosuccinic acid is going to the alpha ketoglutarate acid. Okay, see 6 carbon to 5 carbon, 1 carbon dioxide is released over here. Okay, now this one, okay, this oxalosuccinic acid over is the carbon dioxide release, and here there will be oxidation. So this oxidative decarboxylation. This is the process only here. This carbon dioxide is released and coenzyme is added. That's why this is succinyl coenzyme. Okay, fine. So this is oxalosuccinic acid. There will be carbon dioxide is released. That is called alpha ketoglutarate acid. Now here this alpha ketoglutarate acid again going under the what decarboxylation. Okay, oxidative decarboxylation. Here this one is oxidized to succinyl coenzyme A, and there is a oxidation is happening over here. So that due to the oxidation, this NAD is going to reduce to the NADS. See, in this, why this is oxidation? Where is oxidation? Where is reduction? Now see, in this alpha ketoglutarate acid is oxidized to succinyl coenzyme A, but this NAD is reduced to NADS. See, there is a addition of a hydrogen. That's why it is reduced. When there is a here is reduced, this reaction will be oxidized. Okay, I think you understood. This alpha ketoglutarate acid, this coenzyme A is added because of to stabilize the energy over here. Now again this succinyl coenzyme. This succinyl coenzyme A is this 5 carbon to 4 carbon. Now carbon dioxide is released over here. Okay. Now if you can see, there are from up to this reaction there are 2 carbon dioxide is released. Okay. Now this succinyl coenzyme A, we will remove this coenzyme A. Okay. Now here it is added to stabilize the things. Now here this coenzyme is removed again, and in that process there will be GTP is forming, that is substrate level phosphorylation is happening. Means GTP means this GDP is forming to GTP, and in that process there will be one ATP will be formed. So ADP will be formed to the ATP. Okay, fine. So this succinyl coenzyme will be give rise to the succinic acid, 4-carbon compound, and this is the succinic acid to fumaric acid that I said you the, that is the membrane level one enzyme is there. Okay, so which is membrane based, all are membrane based are the all are the matrix based enzyme. This one is only the internal that mitochondrial membrane enzyme. This succinic dehydrogenase, which succinic change to succinic acid to fumaric acid, followed by the oxidation, and this here what happened, there is a different compound FAD to FADS. Okay. Now this fumaric acid to malic acid, then malic acid to oxaloacetic acid. You remember that oxaloacetic acid is reduced to malic acid in the C4, but here malic acid is oxidized to oxaloacetic acid. Here NAD to NADS. That is malate. Wherever there is a, I said already before in the time of photosynthesis, where there is a reduction, that is a dehydrogenase. See, a dehydrogenase, here dehydrogenase, here dehydrogenase, here dehydrogenase. Okay, fine. So these are the thing. Okay, now we will just go through the lines of what is saying that citrate is then isomer to isocyclate. The TCA cycle starts with the condensation of acetyl group with oxaloacetic acid and water to yield citric acid. The reaction is catalyzed by the enzyme citrate synthesized with a molecule of a coenzyme released. Okay, now you see citric acid, fast reaction, put it into one oxaloacetic acid, acetyl coenzyme is added over here from the link reaction on the presence of citrate synthesis. Okay, and water is there, so that is a condensation reaction, and it will go to the citric acid 6-carbon compound. Now what is saying? This is the first step. Citrate is there isomerized to the isocitrate. Now see, this is the citric acid. Okay, this is the first reaction. This first reaction is going to second reaction, isocitric. Okay, this is isomerase. Okay, isomerase activity is happening. This is followed by the two successive states of a decarboxylation leading to the formation of a ketoglutaric acid and a succinyl coenzyme. Now it is followed by two successive steps of a decarboxylation. Now what is saying that there is two successive steps of a decarboxylation. Now see over here, this is 6-carbon. This site that isocitric acid is going to the what oxalosuccinic acid. That is the one reduction oxidation is happening. There is one here is NAD is reduced. Now here see there is a two successive decarboxylation. One is here, one carbon dioxide, and another is here. So that is two successive decarboxylation. Okay, fine. To form and then succinyl to alpha ketoglutaric acid and then succinyl coenzyme. Alpha ketoglutaric acid, this one, and then succinyl coenzyme. One decarboxylation is formed the alpha ketoglutaric acid. Again this oxidative decarboxylation forms succinyl coenzyme. Okay. Now in the remaining step of citric acid cycle, succinyl coenzyme now is oxidized to oxaloacetic acid, allowing the cycle to continue. During this conversion of a succinyl coenzyme to succinic acid, a molecule of GTP is synthesized. This is a substrate level phosphorylation. In a coupled reaction, GTP is converted to GTP with a simultaneous synthesis from the ATP from the ATP. What happened in this succinyl coenzyme? What happened? This is formed when it is forming the oxaloacetic acid. There is in the first step of when it is forming the succinic acid, this coenzyme is removed, and there is a substrate level photophosphorylation is followed by the formation of a GTP. This GTP can be what is saying that it is saying that it can be converted with a simultaneous synthesis from the ATP to ATP. So this formation of GTP is that simultaneous. It is similar to the formation of one ATP over there. Also, there are 3 points where the cycle where NAD is reduced to NADPH. Okay. Now you see over here. Sorry. How NAD is reduced? See over here. One at here is okay. Here is 2, 3, and here is the FADS2. Okay, that's what he's saying. What it is? Sorry. So Also, there are 3 points where the cycle NADH is reduced and 1 point where FAD is reduced to FADH2. Now, here is one NAD is reduced to NADH. Here is from the isocyclic to oxalosuccinate. Now, one is alpha ketogluteric acid to succinyl coenzyme. Another is malic acid to oxaloacetic acid. This is 3 NADH, and here is one FADH. Okay, fine. Where it is? Okay. The continued oxidation of acetyl coenzyme via the tricarboxylic acidic cycle requires the continued replenishment of oxaloacetic acid, the first member of the cycle. In addition, it also requires the regeneration of NAD and FAD place from NADH and FADH2 respectively. Okay. Oxidation or dehydrogenation occurs at 4 places. Now see what is it? Oxidation or dehydrogenation occurs at 4 places in one Krebs cycle. Now see where it is happening. In one cycle, there are 4 places where the oxidation or dehydrogenation is happening. Now see here, oxidation where is happening. One step is here, okay, fine. Another here, another this one, 3, this one, 4, okay. Formation of what? 3 NADH and 1 FADH2, along with 1 GTP or 1 ATP produced by the substrate level phosphorylation in each turn of it inside. So there are how many ATP? So there is 12 ATP. Okay, yes or no? बुझिपाल ना नहीं केमती 12टा ATP हला देखो 3 NADH will give to the 3 into 3 9 ATP. Here 1 FADH will give 2 ATP. So 9 to 11. Now here 1 ATP will be 12. Understood? Yes or no?
Yes, sir. Yes, sir.
Link reaction and Krebs cycle occurs 2 times during the complete oxidation of one hexose molecule. That is, what is a link reaction and a Krebs cycle occurs 2 times. Okay, to oxidation to follow the hexose. And I see in case of if you will go to the this glycolysis, 2 pyruvic acid is forming. Yes or no? So one pyruvic acid cycle we have seen now. So how many to form to complete oxidation of one glucose? Now there will be 2 cycle of a Krebs pyruvic acid going to the presence of oxygen. It will be followed. Yes or no? So 2 means 1. It is forming 12 ATP. So in 2, how many forming? 24 ATP. Yes or no? So
Yes, sir.
So link reaction and Krebs cycle occurs 2 times during the oxidation of one hexose molecule because by the glycolysis one hexose convert into 2 pyruvic acid and both the molecule undergo separate link reaction and a Krebs cycle. Now see in the EMP in the first glycolysis what happened? ATP synthesis to the substrate level phosphorylation over here. Okay, EMP pathway we will study. Okay, this is glycolysis. Okay, fine. So here what happened? 2 and 2, 4. Okay, 4. This is 4. Okay, 2 is already being used. So net is 2 over here. Now 2 NADPH. Now we are studying aerobic respiration. Anaerobic respiration, this 2 NADPH is being used in the formation of ethanol or lactic acid. But in case of when it is going to the mitochondrial matrix, this 2 NADPH will give how many ATP? 2 into 3, 6. So here 6 ATP plus 2 ATP in glycolysis. Now just I'm going through in just a minute. Okay. Now if we'll go through in glycolysis, how many ATP? 2 NADH, yes or no?
Yes,
2
sir.
NADH plus how many ATP? 2 ATP, yes or no? 4 ATP actually, but 2 ATP is being used in the process. Now, in case of what is link reaction, yes or no? So in case of a link reaction, tell me.
2 NAD.
Now glycolysis done. Now in link reaction, what is happening?
So the 2 NADH.
2 NADH. Okay. Now 2 NADH equal to how many ATP?
6 ATP.
Okay. in this glycolysis,
The 8 ATP.
3 into plus 6, 2 into 3, 8 ATP total. Yes. Okay. Now, in cycle,
The 6 NADH.
Krebs cycle, 2. How many NADH is forming? 3
6
over here.
NADH. 3 NADH.
Okay, so 3
So,
into 3 means here it is 2. 11, 1 here, ATP. 12, 12, 2 times it you have to go. Yes or no? 2 times
yes,
1
sir.
glucose oxidation, that will be 2 times. So 2 into how many? 12 ATP. No,
24 ATP.
24, 24 ATP. So total how much?
38
So,
ATP.
so total equal to, okay. So 38, 8, 6, 24, 24, 6, 30 plus 8. Okay, understood? So there are some concepts where generally this 38 and 36 there will be concept is there. Okay, generally this 38 is generally even used at the your what long short kidney. Okay, but in neuron there will be 36. There will be this NADH, one NADH will be there is a concept there. I will take you to the next class. Okay, so there are 38 ATP is generally been net gain. Okay, and in case of a substrate level phosphorylase, as it in ATP gain to the oxidative photophosphorylation is 34 or 32. Okay, so these are the ATP synthesis to substrate level phosphorylation. There are 6. Okay, there are 4 in there in the glycolysis pathway, and in the Krebs cycle there will be 2. Okay, so these are the things. The Krebs cycle, how to means one. This is two. This is only one cycle. If you do second two cycle, there will be here this SLP substrate level phosphorylation 180 with then one other one or two. Okay, fine. I think the concept is understood. This Krebs cycle, or any doubt? If you just you can clarify over here. Any doubts?
না স্যার
ବୁଝିପାରିଲ ତ?
ଆଜ୍ଞା
এইগুଡ଼ା question ଗୁଡାକ କରିବ ହେଲା କେତେଟା ATP means the question can be asked that if one how many ATP will be released from one pyruvate, then you should don't have to follow that one glucose means glycolysis you don't have to follow. So there are many net this that what balance sheet, if you will go through the balance sheet, there are many questions can be asked over here, where what is the first enzyme is being used, cytosine, what is the location of this place. Okay, this succinate dehydrogenase is present in the inner membrane. These are some important points where you have to go through. Okay, fine. Now electron transport chain we will study in the next classes. Okay, fine. Hope
ଆଜ୍ଞା
you understand the thing. Okay, sorry, I'm extremely sorry for my health issues, and from 24 I will be a bit traveling, so these 2 days I will try to this complete this. Tomorrow I will try to complete this chapter. Okay, so let's see what can be done. Okay, so that 24 25 I think I will be traveling to somewhere. Maybe I can take the class, but uh, I will try to take the class in the morning 24 if it is you people allow me to take the class in the morning. Otherwise, tomorrow I will take class 24 morning. If you people are available, then I can take the class. Otherwise, I will be board the train on the 24 means around 12 o'clock or 2. So accordingly, I cannot take the class in the evening. So morning, if you people allow, then otherwise I will record it and same as I was post it into the YouTube so that you can get the thought. Otherwise, tell me the what time you are available on the 24th so that I can go through. Okay, text me over the Telegram. That's all for today. Thank you.
Thank you, sir.